Hey, welcome and thank you for joining our first Virtual Innovation Summit. We have a truly global audience of users today dialing in from across the world. And, in today's unprecedented times, it's really required acceleration of us to come together as a life sciences community to address the impact of the pandemic and other health crisis. Thermo Fisher has been a proud partner with key thought leaders and innovators who will be presenting their progress they've made over the different life science application areas in this PowerPack 3-day event. This keynote is slightly different than normal. It's been more of a conversation around a few topics with some of my colleagues who I'd like to introduce. So today we'll be talking to Dr. Andreas Humer, who's the Senior Director of Life Science Omics, Brad Hart, who's our Director of Clinical Research in Life Sciences Mass Spectrometry, and Steve Millian, who's part of our Viral Vector Services group, the Pharma Services team. So we'll be having a conversation with these three colleagues during the journey here through this presentation. Innovation has been the heart of the mass spectrometry business, producing high-performance systems and very key milestones. Even though this map goes back to 2005, obviously Thermo Fisher was innovating in mass spectrometry for many, many years before that, whether it was in single-stage quadrupoles, vector instruments, 2D, 3D ion traps, FTMS. The Orbitrap was really an inflection point in terms of the major applications that were enabled use that technology. So if you hark back to 2005, which is now 16 years ago, the first LDQ Orbitrap mass spec was really the first system that allowed comprehensive single-shot analysis of, say, the bacterial proteome. Over the next 6 years, we developed a number of systems raised around that technology. But then in 2011, I think one of the most impactful launches was the Q Exactive, which is the first benchtop MS, hybrid MS, for top-down and bottom-up proteomics, and really has significant impact. And you can see the different figures of merits there in terms of the science it enabled. I think importantly in 2013, the Orbitrap Fusion Tribrid mass spectrometer was launched. And this was really a quantum step in terms of throughput and improving throughput, the sample multiplexing, along with things like TMT reagents, which we'll talk about later on. Subsequently, there was a number of other tribrids launched over the next 7 years, as you're well familiar with the Orbitrap Lumos and more recently the Eclipse. We also tried to enable the Orbitrap to enter new market spaces. So the UHMR was launched in 2019. We realized a platform to gain structural insights. So we extended the mass range of that system to 80,000 M/Z, which allows us to look at intact proteins, and really enabled the whole area of native proteomics to become a more accessible technology and application area. Not forgetting small molecules, in 2019, we also launched the IDX Tribrid MS, and we'll talk some more about that a little later on, which is really a tailored tribrid system for looking at small molecules for doing structural analysis in different types of applications, whether that's in scientific research or toxicology or metabolomics. And then, of course, over the last 2 years, we launched the Orbitrap Exploris mass spec family, the 120, the 240, and the 480, which really enabled high-speed analysis and adoption into new application spaces, whether that's in the more applied areas, environmental, toxicology, food, and then really opened up the proteomics and metabolomics space because of its speed and resolution and sensitivity. I think what's stunning is always this slide, which we've shown every year, is the amount of publications that come out of the Orbitrap family. So in the last 12 years, there's actually been over 75,000 articles published using the Orbitrap technology. It's truly amazing. So on the left-hand side, you can see all the journals, and you can then on the right, you can see the pace of publishing of those papers. But this last year, if you really put the math on that, it's 1.5 articles are published every hour, which is truly stunning for that technology and the impact it's had on the industry and the community. I think if you reflect on Thermo Fisher Scientific during this recent pandemic, we have a number of other technologies that have played a role in the pandemic and understanding of the COVID-19 virus. And here's a busy slide, but It shows how our cryo-electron microscopy and also our mass spectrometry have come together to solve different problems. And that may be looking at spike structure, maybe mapping recombinant spike glycoproteins, binding assays for targets, antivirals, structural conformation, glycosylation. And you can see the map here. So a significant contribution from the community in terms of understanding both structure, but also in terms of developing therapies for the disease. So thinking about virus structure, host cell interaction, virus cell and machinery, disease mechanisms, and vaccine development has been a very, very important part of the work that's been done in Thermo Fisher Scientific with the community. I think in addition to the effectiveness of current drugs and biotherapeutics that are assessed along with immune system response, prognostic, phenotyping, and direct rapid virus development, on average, a publication submission was made approximately every other day on COVID-19 research with our very own Orbitrap mass spectrometers. And you can see that in this graph here. These are all peer-reviewed journals. And like I said, if you look at host cell interaction, disease mechanism, drug development, virus detection, virus cellular machinery, you can see the distribution of papers that were published throughout the last year using mass spectrometry for COVID-19 studies. With that said, let's dig deeper to understand the role of mass spectrometry in the pandemic. At this point, I'm pleased to introduce Andreas Humer, our Senior Director of Omics and Chromatography and Mass Spectrometry, a division of Thermo Fisher Scientific, who will begin this discussion. So Andreas, a question for you. Mass spectrometry is a powerful tool for infectious disease research, but really is not often thought of the mainstream technology in this field. From your perspective, how has the role of MS evolved during the pandemic?
Yeah, Iain, thanks for having me. The instant response to the COVID-19 challenge from the scientific community that we witnessed was something I'd never seen in my career before. The entire field pivoted to apply their field of expertise and their resources to answer the critical questions that we needed to respond to the pandemic. The Omics and the mass spectrometry community was no different. From a scientific perspective, that meant we had to quickly understand the structure of the virus, particularly the spike protein that enables the access of the virus to the human body. Weeks after the threat was identified, the first structures were published using cryo-electron microscopy and mass spectrometry, characterizing the important viral structures that ultimately enabled the successful creation of a vaccine that's now being rolled out worldwide. But almost in parallel, however, labs across disciplines collaborated to get a deeper understanding of the biology of infection and the host response. Again, mass spectrometry was a key tool to study the interactions of viral proteins with host proteins. And now we have several research publications that describe how the virus proteins interact with the host proteins to target innate immunity regulators and interrupt potential vital protein phosphorylation signaling pathways to downregulate type 1 interferon response from the host. So in those research efforts, many potential drug targets were discovered, and the work now offers the opportunity to repurpose some of the existing drugs for antiviral therapeutics. I believe there are now over 130 potential drug candidates in clinical trials. And again, this very deep insight to the biology of COVID-19 was made possible by the enormous discovery power of Orbitrap high-resolution mass spectrometry. Now, from a technology solution provider perspective, the COVID-19 crisis has challenged organizations like Thermo Fisher Scientific in ways we had never anticipated. We had to respond quickly to support vaccine development and manufacturing through mass spectrometry. Iain, I know that we had to specifically develop tools for that.
Yeah, that's correct. It was really exciting to see the application of mass spectrometry using our Orbitrap HRM instruments. But the story does not stop with our instruments. It really extends to our software solutions as well. Our Thermo Fisher Scientific Biopharma software 4.1, which we'll be talking about this next week, has the capability to analyze digested mRNA automatically by identifying each mRNA digestion component based upon in silico digestion. So it allows in silico digestion of large mRNA. There's automated component and modification detection, automated MS/MS annotation for all identified components, and visual mapping tools provide percent sequence coverage. These new capabilities combined with our HRM mass specs are providing an invaluable tool for mRNA research in the past year. And this new mRNA mapping capability is the first to market solution for LC-MS analysis. So a very, very important combination of both mass spectrometry hardware, but also software to solve a very important problem and look at new therapeutic modalities. Further downstream, one of the most widely used methods, as you all know, is to detect acute viral infection is a diagnostic real-time polymerase chain reaction, or RT-PCR. However, because of the COVID-19 pandemic, mass spectrometry proteomics was actively being discussed as a potential diagnostic method for viral infections. And although proteomics is not yet applied in routine virus diagnostics, there is huge potential for it to be applied to detect viral infections. Let's learn a bit more about this from Brad Hart, our Senior Director of Clinical Research and Toxicology in the Chromatography and Mass Spec division. So Brad, a significant percentage of the publications are related to direct COVID identification and mass spectrometry. What has led our customers to pursue COVID ID using mass spectrometry? What have we learned about the future of mass spectrometry and infectious disease testing and clinical diagnostics?
Thank you for the question, Iain. In the last year, Thermo Fisher received over 40 requests to collaborate on COVID ID testing by mass spec. Everything from compact sniffers to large-scale lab-based automation platforms. The NIH's RADx proposal kicked off a rush of innovative ideas, primarily around accurate, fast, easy to use, and widely accessible testing in a variety of matrices. However, some of the innovation proposals actually tapped into existing clinical logistics chains using nasopharyngeal swabs and then deploying mass spec as the detection technology. As stated and published by Dr. Pandy of Mayo Clinic here, in Europe, the COVID-19 mass spec consortium assay was considered by the UK NHS as one alternative to deploy the use of mass spec on a large scale. And other parts of Europe were also involved with the COVID-19 mass spec consortium in this regard. So why was mass spec so heavily pursued for COVID-19 ID? For one, it is an alternative supply chain to PCR, and the PCR supply chain was stretched at the height of the pandemic. Also, since mass spec is routinely used in clinical labs, and the COVID testing by PCR reduced the usage of those instruments towards their traditional mass spec test, a capacity opportunity existed in the labs. And COVID-19 mass spec assays were primarily focused on the identification of the nucleocapsid protein. Proteins are stable analytes. It's an attractive attribute for a robust clinical assay. The preparation costs are similar or less than PCR reagents. And as a leader in proteomics, as Iain mentioned, with over 70,000 publications on the Orbitrap, those included viral studies on SARS-CoV-1, MERS-CoV, and other infectious disease, it comes as no surprise that our customers would quickly recognize the potential for rapid identification of the COVID virus with mass spec. 16 papers were accepted in the last year on COVID-19 identification by mass spec. We know that the selectivity of mass spec is unsurpassed, and we verified in this effort that the sensitivity can be equal to PCR, even at low viral loads. We confirmed throughput can be as fast as a sample per minute or better. We know that it is straightforward to multiplex analytes with mass spec towards larger disease panels. And the interest in mass spec being closer to the patient grows yearly. It's still a major challenge as mass spectrometry is considered a complex technology for clinical labs. So where do we go from here? Where do we go next? In the last year, our customers have demonstrated the ability to pivot and rapidly develop mass spec assays for infectious disease in response to the COVID-19 crisis. The work done in 20 and 21 builds on the foundation to effectively apply mass spec to infectious disease identification, as well as provide the capability for phenotypic prognostic information for treatment stratification. A mass spec also offers fast, selective potential for diagnosis of infectious disease panels, which do have added utility for clinical diagnostics. We can imagine a single analysis for the detection and confirmation of common cold, influenza variants, COVID variants, and even tuberculosis. And I'll just say that tuberculosis being preventable and treatable, it still infects 25% of the world's population and is one of the leading infectious disease killers in the world, taking the lives of 1.6 million people each year. So we will continue to develop and expand protein assays towards the clinic in support of lab-developed tests with capillary and analytical flow rates as fully integrated workflows, and even towards rapid analyzers. We will continue to develop and expand IVD-approved mass spec platforms. We registered our triple quads in 20 countries last year, and we'll continue to develop mass spectrometers that can be deployed closer to the patient. And we truly see infectious disease identification and confirmation as a critical opportunity for the mass spec community to make the world a healthier and safer place. Thank you, Iain.
Okay. So thanks, Brad, for that insight. That was very helpful. So along with our partnerships with the outside scientific community, we have also partnered with internal groups inside Thermo Fisher Scientific to optimize services for the pharmaceutical industry. I'd like to introduce Stephen Millian from our Pharmaceutical Services Group and how they've worked with their clients to optimize their viral vector services. So Stephen, I have a number of questions here. Can you discuss cell and gene therapy overall mission around offering LC-MS-based services?
Great. Thank you, Iain, for that introduction. Really, the overall mission around offering LC-MS-based services for our clients is really about just utilizing the full potential of LC-MS technologies to help solve some of the difficult challenges that the industry is facing when attempting to characterize these very large, complex viral vector drug products. Also, as part of this mission is to help further standardize analytical methods used in the industry via publications and the commercialization of assays and kits.
So what do you think our current clients' assay challenges are?
Yeah, that's a great question, Iain. I think some of the greatest challenges that we and our clients face with regards to assays is really around developing accurate, precise methods for characterizing our analytes of interest. The industry is really currently trying to leverage proven analytical methods, including mass spec, which were widely used in the biologics industry and in the mAb industry for the last several decades. I think another big challenge we face is with the consistent supply and quality of reagents and standards. And lastly, there's a considerable challenge around data integrity and better CFR 21 Part 11 compliance when it comes to instrument software and data analysis.
Okay. My final question is, how did your team internally accelerate their offering to adapt to specific client needs?
Yeah, as part of overcoming some of the challenges I just mentioned and doing it in an accelerated way has been to make strategic investments in the latest LC-MS technologies. We've also partnered up, as you mentioned earlier, Iain, we've partnered up with other internal groups within Thermo to try to develop novel approaches for characterizing viral vectors and process-related impurities.
So, Steve, what was the downstream impact to our clients?
Feedback thus far from clients has been extremely encouraging. Our clients also see the advantages in leveraging these very powerful analytical methods. We now have the ability to use the most advanced instrumentation and analysis to better characterize our clients' drug products. We are also exploring ways to provide products such as SureQuant kits to further standardize and streamline assays for viral vector products. This will have a huge benefit for our clients as it will dramatically reduce the time needed for assay development and inevitably will also reduce the time needed for release of drug batches.
Yeah, thanks, Stephen. That's a powerful example of how mass spectrometry-based proteomics is having a direct impact in the analytical sciences area. The Shotgun technology and deployed in your example, came out a major advance in the proteomics field and has been driving the past decade. So a nice example of how we've been able to find an adjacent application in the biopharma area. So the other area that's capturing researchers' imagination is around single-cell proteomics, which seems to be on the cusp of being accessible to more researchers and not just a niche research application. We know this research area will make significant contributions to cancer research, the ability to look at single cells and study single diversity. So Andreas, I'll bring you back in here. Can you tell us about the progress here and what's happening in the field?
Yeah, Iain, single-cell analysis has been a field predominantly driven by the scalability of genomics technologies. And as you said, non-genomic technologies are now quickly progressing to the point where a meaningful number of proteins can be quantified and studied. So we're talking about more than 2,800 proteins from a single isolated cell. There are several key technologies that are coming together to enable this progress. We will hear about commercial solutions for sample handling in a session later this week by Erwin Schuf. It's not just the ability to isolate single cells, which would be useful in studying rare cells, for example, but also the ability to provide scale and the throughput to study several hundred cells per day. Multiplexing is obviously the strategy that provides the throughput that ultimately enables studies across entire tissue sections, where researchers hope to learn about cell heterogeneity and cellular connectivity in the context of a disease, such as cancer. Multiplexing capabilities and sensitivity of the mass spectrometer at a single cell level are key for significant findings beyond one cell at a time. And that will enable real biological understanding from higher throughput single cell profiling and quantitation to spatial omics. I'm very excited to see how quickly this field is accelerating from the first publication in 2017 from the Slavov lab, identifying hundreds of proteins in non-oocytic mammalian cells using Orbitrap mass spectrometer and the continued growth of applications from the community using Orbitrap mass spectrometers. We are dedicated to driving the progress in single-cell proteomics and making this accessible to everyone. We will do this by partnering with the thought leaders and practitioners from both the scientific community and bringing in our experts in R&D and application teams to harness the insights, innovations from both sides, from ideation to development, and deliver solutions, be it technology, which also includes sample preparation and handling, applications and methodologies for single-cell proteomics.
So we talk about cancer here, and biomarker research has been an area that proteomics community is focused on for some time now. So Andreas, how do you see this MS evolving as a key technology in the area of proteomics, particularly in how new technologies are applied to translational space and in the clinical research applications?
Mass spectrometry is often seen as a specialized expert technology, particularly by scientists in the clinical translational research space, where standardization and scalability are required. In the biomarker field, plasma has been a tissue of choice, and the proteomics field has focused for a long time on providing depth of protein coverage for most complex protein mixtures that we know of, which is plasma. Of course, this has meant in the past that ultimately researchers have to trade off between throughput and discovery power. More recently, however, there has been a rethinking in the field, and researchers are focused on scalability, providing higher throughput with enough discovery power that other technologies cannot provide. We will hear more in our Trends in Life Science Omics Research opening keynote on Thursday from our partners and industry leaders on how we are advancing deep, rapid, large-scale proteomics utilizing Orbitrap mass spectrometer and novel sample preparation technologies. I would say plasma proteomics is crossing the chasm.
So speaking of crossing the chasm, are those advances primarily driven by sample preparation technologies? Are we seeing other technologies emerging to drive those breakthroughs?
Advances in sample preparation are needed to drive standardization across the workflow, and we have launched a number of relevant consumable products in this space over the past few years to help standardization and automation. But in addition to generating more useful data analysis tools have evolving out of deep learning efforts are now providing a complete new level of spectrum analysis that were unthinkable just a few years ago. On Thursday, I encourage you all to join the session by Martin Freno, who will show how artificial intelligence is transforming proteomics data analysis.
So how are the translational efforts progressing in other omics, such as small molecules, i.e. metabolomics?
I think the basic needs for standardization and scalability are pretty much the same in metabolomics, with the difference that standardization of metabolomics approaches for plasma analysis has not progressed as much compared to proteomics. To really help drive this process in the community, we're very much involved in the QC/QA efforts of the community, the MQuack group. We recently partnered with UC Davis West Coast Metabolomics Center to form a Center of Clinical Metabolomics with Professor Oliver Fien to help drive best practices and unlock the power of standardized workflows around the Orbitrap technology in a number of large-scale studies. Over the past several years, we have been working hard to enable the use of mass spectrometry technology to non-expert users. The intelligent mass spectrometry methods that we introduced several years ago are good examples of that and have helped to drive adoption and ultimately democratization in metabolomics. Example here might be the AcquireX data acquisition workflow approach we launched several years ago. AcquireX has enabled non-experts to gain a depth of coverage from complex samples that was not possible before. More innovations around the topic of intelligent mass spectrometers will be coming forward in the future that will continue to drive the democratization in the space of omics.
Thanks, Andreas, for that insight. That was very, very helpful. So I hope you've enjoyed our conversation as much as we have and how we're shaping the future of LC-MS in the life sciences community so far. You know, with our discussions about the Orbitrap-based innovations, I'm sure you're wondering what other innovations we've been working on during the last year during the pandemic. Okay. For small molecule unknown analysis, the primary goals are to detect compounds of interest and understand their structure. You know, the elucidation of chemical structures is one of the most difficult challenges in analytical chemistry. Background and matrix interferences can inhibit the quality results, and manually creating background exclusion layers and matrix features is labor intensive. MSMS is often insufficient for structure assignment. MSN can help, but requires extensive experience to set up. There are also additional challenges about how to best visualize the extensive data sets and confidentially assign matching identifying for an unknown. Now, acquiring more data is one part of that story, but compound identification is the next step for comprehensive identification of all your compounds. So using Thermo Scientific Compound Discover software in combination with a range of spectral libraries such as MZCloud, compound databases, ranking tools such as Thermo Scientific MZLogic can really aid to unknown putative identification. You know, all this provides extensive spectral library fragmentation information and structural databases. So small molecule researchers have high confidence in assignment for more unknown compounds in less time. You know, as for the slide here, Thermo Fisher Scientific developed a comprehensive hardware and software solution to address these challenges. And as Andrea said, back in 2019, we launched the Thermo Scientific Orbitrap IDX, which was a dedicated tribrid mass spec coupled with intelligent workflows such as AcquireX, data acquisition, and Compound Discover as a total solution for small molecule identification to address this identity crisis in small molecule compound ID. Today, I'm really pleased to introduce our next generation of small molecule dedicated tribrid mass spec, the Orbitrap IQX tribrid mass spec. The instrument has been built to help small molecule research capture more low abundance unknown analytes, so they can generate more meaningful data from multiple applications, whether that's lipidomics, metabolomics, metabolite ID, any other small molecule application. As you can see, compared to its predecessor, there's some new features in here, notable improvements. MedIQ data acquisition, which is now a real-time library search, which allows us to do confident identification of structurally similar molecules, so MedIQ. There's also remote calibration with a new order-ready ion source, which allows users to spend less time on instrument setup. It has automated scheduled calibration times. It has its own emitter, its own sample tube to avoid calibration. And then the Orbitrap IQX instrument comes equipped with the enhanced deep AcquireX data acquisition, so enhancing that workflow, which provides a fully automated method to perform exhaustive precursor selection for high-quality MSN data acquisition. So now we can combine up to 5 inclusion lists and exclusion lists. We can submit several experiments from a signal sequence. And as you can see, there are other features in here which are enhancing the capabilities of small molecule analysis. So the two workflows which we've talked about, AcquireX data acquisition, which is an exhaustive methodology for going deep and profiling, and then the real-time library searching, which is MedIQ, for unknown identification based on related compounds. So the two workflows, AcquireX is unknown unknowns, which is a workflow looking at an exhaustive methodology, looking at precursor ion mass, accuracy, MS1, experiments, and then creating user lists for our next experiment so we can go deep into a sample. The MedIQ is a known unknown experiment where we're looking at related compounds, or almost a targeted experiments where we have related compounds but unable to distinguish by precursor mass alone. And basically we create MSN fragmentation of precursor ions, and we automatically search those against special libraries, then trigger further experiments to go and look at substructure of related compounds. Now, both of these workflows can be combined, obviously. You can use AcquireX in a real-time library search. So you can use AcquireX to remove background ions, and we can use a more targeted experiment to go and look at structure-related compounds. So here's the MedIQ intelligent data acquisition flow for its intelligent MS for small molecules. So the challenge here is identifying unknown metabolites, degradants, or transformation products. The solution is an intelligent MS and data acquisition, which enables annotation and characterization of unknown compounds. So you can see the workflow here. Fundamentally, we take the parent drug, a prodrug, which is a known compound, and create a spectral library. And this may be through multiple HCD energies, it may be through different fragmentation modes, and those are stored in the library. We then run the unknown compounds mixture, unknown related compounds, and we compare the MS2 spectra to the library in real time. If there's a similarity match, we actually trigger an MS3 experiment. And we can characterize the unknown compounds using a combination of the MS2 and MS3 spectra. So the workflow is smart. It triggers very few and very selective MS3 scans only, based on MS2 spectra that's similar to those compounds of interest. It's also doubly smart because MS3 are triggered on fragments that are unique to the unknowns, and it's the quickest route to the structural elucidation. So effectively increasing the MS2 scan rate for sampling more precursor MS precursors. So this is a very, very powerful and intelligent methodology to essentially look at compounds with spectral similarity in a complex mixture. And here's just a real example using intelligent MS3 with real-time library search. You can see the spectrum at the top, which is the known drug compound of interest, and then we match the MS2 spectra from the library to the experiment, MS of the unknowns. And you can see there's a few peaks that are slightly different, a lot of similarity. So we trigger MS3, and the spectra at the bottom are the MS3 triggered substructures from the MS. This is a very, very powerful way of digging deeper into spectra and molecules of similarity, whether it's a metabolite or transformation product. And here's a real example. Here's a real example from Genentech. It's a study of Prevnar metabolite identification using the new MedIQ workflow. So MedIQ, with data-dependent triggered MS3, demonstrated 54% more metabolites identified than traditional data-dependent experiments. So the experimental conditions you can see down the side, and comparative results were compared with a set of previously characterized metabolites. MS3 was triggered for 17 metabolite peaks in the MedIQ DDA versus 11 metabolites in the traditional DDA experiment. So the power of this workflow really allows us to go looking at very complex mixtures and find compounds of similarity which would normally be missed because of the background and other factors in terms of the acquisition rates that we run at. If you'd like to learn more about how the Orbitrap IQX is being used for a host of applications, including impurity analysis, MedID, lipidomics, or it could be untargeted metabolomics, don't forget to attend the sessions in the next 2 days. They'll be given by Krista Crittenden and Scott Stanley, Josh Kuhn, and Gary Patty. Will cover a range of different applications using an advanced intelligent data acquisition methodologies. We are also introducing the FAMES Duo Pro interface, which extends the capabilities of our differential and mobility to a broader range of applications and also a broader range of flow rates. The FAMES Pro Duo interface is an orthogonal technology for doing gas phase separations, which reduces background complexity and allows more selectivity in analytical analysis. So the system here is optimal for both low and high flow rates. It now runs from 100 nanoliters to 1 milliminute. The interface is compatible with all our mass spectrometers. It has online CV optimization capabilities, not only for high resolution accurate mass systems, but also for triple quads for SRM type applications. We also have faster ZV switching times for targeted and quantitative applications. So the FAMES Pro Duo interface is really allowing an extension of our gas phase separation capabilities for more targeted applications and ultimately increasing detection limits in very complex mixtures. There's a couple of examples here. Here's an example of a set of sulfide vet drugs spiked into a meat matrix, which are extracted and analyzed. Without FAMES, there's a huge presence of solvent ions and also co-eluting matrix ions. You can see the DSC is a very noisy background. When we introduce FAMES selectivity, we suppress both the abundant solvent ions and the co-eluting matrix ions. And the base peak detected on the target compound, which is actually a 10x fold lower intensity, can now be detected reproducibly and quantified. So the power of this technology to reduce interferences and become more selective for target analysis allow us to get lower and lower detection limits in very complex matrices. And just a second example is a targeted compound quantitation on a triple stage quadrupole, the TSQ Altis, looking for testosterone, and detection really becomes a challenge when extracted from plasma due to the background interferences. This was actually run at 250 microliters a minute. And the example here reduces the matrix interferences to increase the signal to noise, so we have reliable detection of the compound. But also removal interference improves automated peak integration for improved confidence in the method efficiency. So another very simple example where FAMES introduces selectivity to allow us to do high performance quantitative analysis on a triple stage quadrupole. So if you want to learn more about either the Orbitrap IQX Tribrid Mass Spec or the FAMES Duo Pro interface, our product experts will be available for you daily during the set event hours, which you can see on the slide here. They look forward to chatting with you further about these later innovations and getting into more details about the scientific applications that can be addressed with these technologies. So switching gears from LC-MS to GC-MS, earlier this year we launched our Thermo Scientific Orbitrap Exploris GC and GC240MS to extend the capability of ultra-high resolution GC mass spectrometry experiments. You can see here, this is the Exploris GC. This is a full-scan high-resolution system providing accurate mass for a series of different types of applications. The beauty of this system, obviously, is through method development, we spend less time evaluating data, increase accuracy because of the high-res and accurate mass capability that this system provides for a number of different applications, whether that's in the food safety space, environmental, could be in the toxicology area. The second instrument, the GC240MS, is a more advanced system. This system provides an unprecedented 240,000 resolution, which provides the highest data quality to accelerate scientific discovery. This also system has options with flexibility, so we can have direct analysis probes. You can look at the EI, you can look at CI ionization, and MS&S. So it really extends the application space, and also areas such as metabolomics can benefit from the Orbitrap GC240. So proteomics technologies have really been evolving over the last 15 years, providing more sensitivity, more quantitative capability, but also requiring more high throughput. Look at things like protein dynamics and look at very large sample sets. You know, over the last 15 years, the critical advances in instrumentation and also the development of isobaric tandem mass tags have enabled more accurate high throughput quantitative analysis of proteomes. You know, the slide here really shows the journey from 2005, where TMT2, TMT6 came into play, and instrumentation mapping with it as well to enable analysis of those TMT tags. But as you see, as we move forward, we move from 6, we move to 10, we move to 11, we move to TMT16 in 2019. And that really has advanced our capability to do really high throughput proteome analysis and do it very quantitatively as well. Thermo Scientific is, excited, as you can see there at the end of that slide, to introduce 2 new isobaric labeled tags, the TMT Pro 134C and the TMT Pro 134N, that can be seamlessly integrated into our workflows and our current instrumentation and isobaric series. This expands the breadth of isobaric tagging that's available from 16-plex to an 18-plex without sacrificing proteome depth or quantitative performance. Just down on the right-hand side there, you can see a recent publication on the TMT Pro 18-plex, which illustrates how higher multiplexing capability reduces data acquisition time and results in fewer missing values across experiments. Therefore, really increasing efficiency without losing quantitative performance. So a very, very powerful addition to our plexing capability. The TMT Pro 18-plex tags will be available in July this year. We'd also like to recognize the 7th annual Thermo Scientific Tandem Mass Tag Research Award. This award is open to researchers enrolled in doctoral or postdoctoral programs. The top 2 recipients were selected in combination with Thermo Fisher Scientific and Proteome Sciences. The grant is awarded based on innovation and potential impact of the application's proposal of using our tandem mass technology in conjunction, obviously, with other mass spec reagents. The research award is $11,000 and used to purchase eligible Thermo Scientific mass spectrometry reagents and also TMT labeling reagents. And the 2 winners this year, I'm pleased to announce, is Owen Hovey from the University of Western Ontario and Ji Zhang from the University of California, San Diego. So congratulations to both of you for winning this award. So thanks for joining us today. We'll take a short break before the live Q&A, where we'll be excited to answer your questions. And we hope you enjoy the next 2 days as you hear from our life sciences change makers. You can see the schedule here. You'll hear a lot of talks around biotherapeutic analysis and also omics, and also in the healthcare area in clinical research. So before the event ends, don't forget to check out the Speak to an Expert Lounge or our poster hall and our resource hub for all the product and application contents. And we truly appreciate you attending this session today.